Thank you, Anthea. Thanks very much for inviting me to come and talk today. Can you hear me at the back? Yes, okay, good. So what I want to talk to you a bit about today is something which in public health you'll either have used by now or will have heard about it, or I guess will have at least thought about it, I hope, which is genomics, so whole genome sequencing, and particularly related to communicable diseases. So I want to give you some examples today of how we've used genomics for public health action. I guess when we think about public health, or when you guys certainly think about public health, you're thinking, I guess, a lot about the clinical epidemiology, but when we think about public health from a laboratory aspect, we think about public health microbiology, and that's actually a distinct discipline. And also I'll talk a little bit about what that is. I'll give you three I guess, areas where we've used genomics, so in the area of antimicrobial resistance, foodborne disease, and environmental point source outbreaks. And then I'll finally just finish by talking about what I think the future for genomics in public health is. So public health microbiology, I guess, can broadly be defined as a very cross-cutting area. So it's, when we think of clinical microbiology, we sort of think of little hospital labs that process urine and poo for patients in hospitals. But public health microbiology is a lot more, I guess, diverse than that. So we receive isolates from humans, we receive isolates from animals, from food, and from water as well. So it spans a variety of disciplines as well. But I guess the predominant focus is on human health and disease. And what it really requires is the ability to work across a number of disciplines, whether it be epidemiology, whether it be public health and policy, whether it's academia, whether it's with clinicians. And I can tell you right now, it requires a lot of diplomacy, a lot of time on the phone, and yes, still a lot of time with Excel spreadsheets. And so I would say that public health microbiologists have a really kind of central role in surveillance and outbreak response, and providing an evidence base for the prevention and control of communicable diseases as well. So when we think about public health laboratories in Australia, I guess it's not really an elephant in the room, it's just a statement of fact, I guess, that Australia does not have a CDC. So there's no national CDC in Australia, but what we have is a network of public health laboratories across the country, and also a variety of structural models which can pose challenges, I guess, in responding to outbreaks in Australia. So there are four main structural models of public health microbiology services in Australia. One is actually for a jurisdiction just not to have one and to work with larger laboratories, and I guess that's for some of the smaller jurisdictions. Some jurisdictions have hybrid clinical and reference laboratories, whereby they have a diagnostic component in the laboratory, but they also have a reference function as well. And so that would be New South Wales, for example, or South Australia, or WA. The FSS, the Forensic and Scientific Services in Queensland, that's actually, I guess, part of a government department. So it's a government organisation. And then Victoria here, so MDU, Microbiological Diagnostic Unit, or you can see why everybody calls it MDU because it's a mouthful. It's actually a little bit quirky in that it's one of the few public health microbiologies globally, in fact, to be part of a university department. So it's part of the Department of Microbiology and Immunology at the University of Melbourne. And so these public health laboratories around the country are coordinated through something called PHLN, so the Public Health Laboratory Network. Why am I telling you this? Well, it's actually important when you think of national responses to outbreaks. So you can see that coordinating different jurisdictions with different models of service delivery for public health, it's not a homogeneous thing, it's different across each jurisdiction. So MDU is now located in the Doherty Institute. So the Department of Microbiology and Immunology is in the Doherty Institute. It's in Parkville. We moved there in September 2014. But MDU is actually the oldest public health laboratory in Australia. It was established in 1897, and it came out of a royal commission looking at the state of Melbourne's water. And it was found, actually it wasn't the water that prompted MDU to be formed, it was the fact that typhoid bacilli were found in beer in Melbourne. And so that apparently was unacceptable. The water doesn't matter, but that was unacceptable for the beer not to be drinkable. Anyway, and so MDU was formed, and that's the original contract there between the Department of Health and MDU. And we provide state-level public health microbiology services, some national functions and some international functions. So we're a WHO regional reference laboratory for invasive bacterial pathogens. What we've been doing at MDU, I guess, over the past 5 years, I think it's safe to say now, I've been taking methods that we use in the laboratory. Some of the methods we've used for 100 years, not the same staff, there has been some staff turnover, but so some methods like serotyping or I guess running gels, even just looking at bacteria on plates and identifying them biochemically, we're transitioning all of that onto whole genome sequencing. So and I'll explain a little bit about what whole genome sequencing is shortly, but really consolidating all of the microbiological methods that we use in public health into one, I guess, one-stop shop assay, so whole genome sequencing. And that has raised fantastic opportunities, a lot of challenges, but on the whole, the benefits of that have been fantastic. We have an in-house bioinformatics team now at MDU, so this has really been one of the major, I guess, growth areas in public health microbiology, bioinformatics. So we have a team of 5 full-time bioinformaticians now to analyse all of the genomic data that we generate for public health purposes. And they take that genomic data and turn it into something that we can interpret as public health microbiologists, and then that we provide back, and so that you can action. So what do I mean by whole genome sequencing? Well, I guess there are a number of, this is a very simplistic diagram, but I think it illustrates the point. So we can conduct sequencing on a gene, so one gene in a bacterial genome. We can sequence that and have a look at that. But if you think about it, so for example, the Staph aureus genome is 2 million base pairs. And if we look at a gene, it can be anything between 400 and 1,000 base pairs. So that's a fraction of the entire genome. So we can get a little bit of information, but not a huge amount of information. We can do the entire genome, so that would be sequencing every single nucleotide, so the A, C, G, T in the genome. So we can sequence all of those. When we do and you may have heard this term called metagenomics as well. That's distinct from genome sequencing. So metagenomics would be where you take, for example, a clinical specimen, you take a swab specimen and you sequence everything that's in that, and that includes the human DNA as well. So that's what we loosely think of as metagenomics. So whole genome sequencing is generally where we have a bacterial isolate or a viral isolate, and we sequence the whole genome of just one pathogen, and metagenomics is where we sequence everything. Does that make sense? Or yeah, okay. So whole genome sequencing, where we look at the entire content of genetic material. And when I say entire content, that's not just the chromosome, but if you cast your mind back to high school or medical school for entry level, you can remember that bacteria don't just have a chromosome, but many of them have mobile bits of DNA that they have as well, so plasmids and things like that. So it's sequencing everything there. So what we do, once we've sequenced everything, we align everything up. So we align all of the nucleotide sequences here. We apply some mathematical models to them, and we come up with a phylogenetic tree, which we use to look at how related things are to each other. And this is, I guess, what a phylogenetic tree might look like. And this is a slide. Some of you, if you've ever been to a genomics talk before, there's a quite famous slide that does the rounds, which Torsten Seaman, one of our bioinformaticians, put together. But it's used as a sort of 101 of how to interpret a phylogenetic tree. And so you can see here that essentially the longer the lines are here, the further things are away from each other, right? And the closer they are, the more related they are. So you can see that this long line here indicates that the banana is quite some distance from its nearest neighbor, this being the mylobar, and is quite a distance from the Freddo frog, okay? But these two Freddo frogs, there's no long line here, so they're essentially the same. This Freddo frog looks the same, but there's a little bit of a line here indicating that there's some difference there. This is actually the mutated UK version of the Freddo frog, which has about twice as much sugar in it. So that's just to illustrate the point. I know it's simplistic, but it is a good way. If you've never come across this kind of data before, it's a good way of just getting it in your brain to start with. All right, so let's think about some case studies here. So I'll talk a little bit about antimicrobial resistance. And what I want to talk about here is sexually transmitted shigellosis, because this is a very recent thing that we've been working on, but really highlights, I guess, how we've used genomics to look at not just an important public health problem, but also antimicrobial resistance. And so shigellosis causes a major global burden of diarrhea. And I guess globally the biggest burden still remains in low-income countries in pediatric populations, and that's where the biggest burden of disease is globally. And there are two main species, Shigellosis-summary and Shigella flexneri. One of the important, I guess, features of shigellosis is that it has an extremely low infectious dose. As little as 10 bacteria can make you very, very sick. So it's extremely infectious. And it's one of those, I guess, unusual causes of gastroenteritis insofar as it's one where treatment is often recommended. So as you will know, most gastroenteritis, self-limiting, doesn't require treatment, but shigellosis is one where the WHO actually still recommend treatment, with the first-line treatment globally recommended as ciprofloxacin, with azithro and cotrimoxazole being second-line treatment recommended options. There are two key features that have changed with regard to the epidemiology of shigellosis over the past decade. One is that in high-income countries, in urban settings, shigellosis has really changed from being, I guess, a cause of sporadic gastroenteritis to being an STI. So certainly in urban Melbourne, endemic shigellosis is amongst men is essentially an STI. The other thing that has changed as well is the way that people travel and where they travel to, and importing their Shigella back into, I guess, higher income countries. There are more imported cases of shigellosis, and that was described by, actually this was, Courtney Lane's an epidemiologist with us at MDU, and she did that for her MA project about 6 years ago now. And the other thing that's changed with regard to the epidemiology of Shigella is antimicrobial resistance. So rates of resistance in Shigella globally have increased quite markedly. And so much so that there was a health advisory put out by Brett, the Chief Health Officer in Victoria, a couple of years ago. It was actually updated earlier this year, and I know that one went out from New South Wales last year as well, just saying that really be guided by the susceptibility results here, don't just treat empirically. And so globally there have been some case reports described of Shigella being transmitted through MSM, just smaller case reports of MSM transmitted Shigella being very resistant to antibiotics. The incidence of shigellosis has actually increased markedly in Australia over the past 5 years. Two reasons for that. One is a change in testing. So formerly Shigella was diagnosed, and actually generally in many labs is still diagnosed through culture. So feces will come into the lab and it's cultured in an agar plate, and we look at it and go, okay, that's probably Shigella, and we identify it. But a lot of labs have changed to PCR-based diagnosis now, so the fecal samples will come in, and instead of being cultured, they'll undergo PCR, and it's detected that way, and that's a lot more sensitive. Therefore, you can see that there's an increase there. However, we know that there has actually been a true increase in the incidence of shigellosis because we're getting more and more cultures as well. And you can see that the incidence has gone up more in males. And so we were interested at MDU. So being the state public health lab, we get every single Shigella isolate in the state for characterization. And so we decided to look at our Shigella isolates from the beginning of 2016 up until around mid-2018. We thought we better have a look and see our isolates becoming more resistant. And when we looked, excuse me, we were absolutely horrified to see that the azithromycin resistance rate in Shigella flexneri and Shigellosis-summary was about 50%. And when we looked at the literature, that was by far and away the highest resistance rate in Shigella that had ever been described. And so that was a concern to us. Nothing else of that magnitude had been reported before. So we performed whole genome sequencing on all of those Shigella isolates. It was about 540 odd isolates, mostly Shigellosis-summary. And this is the beauty, I guess, of having a good relationship with your Department of Health. So we worked very closely with the Victorian Department of Health to link up the genomic data and the epidemiological data. We published on that earlier this year as well. And so what we found, I'm just going to talk through the Shigellosis-summary data because the Flexneri story is very similar. So what we found were 1, 2, 3, 4 major lineages or clades of Shigellosis-summary. And if you remember, I told you about the lines. The longer the lines are, the more distant things are from each other. And you can see that this clade here, there are hardly any lines here. And what does that mean? Well, that means that they are highly related to each other. So they're the Fred of frogs that I talked about. They're very, very related to each other. And when we look at a tree like that, we think, okay, well, that's like an outbreak. That's almost like a point that's either point source outbreak or very kind of dense person-to-person transmission. Yep. So that it doesn't have much time to evolve. Then when we overlaid the metadata onto this, what we found was that these 3 lineages here were highly associated with travel. Yep. So these were the imported cases. And interestingly, this one here, what this actually provided was a sort of geographic framework, because the ones in the purple were associated with travel to India, the ones at the top were associated with travel to the South Pacific, and then the orangey ones were more associated to travel with Africa and the Middle East. But you can see there, the ones that are highly related to each other were almost exclusively from the MSM community, indicating not point source outbreak over 2.5 years, but very dense person-to-person transmission. Then when we looked at the antimicrobial resistance data, I won't go into this in too much detail, but this is the key thing here. So azithromycin resistance was very strongly associated with the MSM population. And one of the key features here was that the azithromycin resistance was mediated by a plasmid that contained a gene coding for azithromycin resistance. Why is that important? Well, when we were doing this work, actually, Kate Baker and Nick Thompson from the UK published on this at almost exactly the same time as us, doing a very similar study in the UK and France. And they described a plasmid with a gene encoding azithromycin resistance on it. And so this is one of the beauties of genomic data, is that it's sort of universal. So you can transfer genomic data across the world in a matter of minutes. And so we were able to share data with them and compare that plasmid that I talked about with their plasmid. And sure enough, again, I won't go through this in a huge amount of detail, but it was essentially the same plasmid. Yep. And so this is a global outbreak of a multi-resistant plasmid, not in Shigellosis-summary, actually in Shigella flexneri as well, so crossing different Shigella species, which was caught almost exclusively in MSM. So what do we have here? Essentially, we've got a perfect storm, I think, and from a public health perspective. So we've got these very dense transmission networks amongst MSM. We've got a pathogen with a very, very low infectious dose that harbors this very multi-resistant plasmid. And why, if you think bigger picture, why would azithromycin resistance be higher amongst the MSM community? And I think what you need to think about is this a population that is exposed to a lot of antibiotics? And in general, it actually is. And when you look at empiric guidelines for the treatment of urethritis, for example, and treatment guidelines for gonorrhea, so for about 5 years now, what's been recommended is dual therapy with ceftriaxone and azithromycin. So this is a group that is under immense selection pressure from azithromycin. So you can see that all of these things, you've got host, pathogen, and environment here, your epidemiological triad combining. So what we've shown here using genomics, I think, are these distinct epidemiological contexts for Shigella in Australia, MSM in urban areas, travel related. Actually, one which I have not touched on, but some of you will know that there's a large Shigellosis outbreak going on, actually in the top end of Australia amongst Indigenous communities. And we didn't look at that as part of that study. We've got this global plasmid outbreak of multi-resistant Shigella, plasmid that crosses lineage, species, and borders. And I think the success of that is facilitated by azithromycin use. All right, so that's an example, I guess, of how we've used genomics to look at antimicrobial resistance. I want to talk a little bit now about foodborne disease. And there are many ways that we can use genomics in foodborne disease. And so one of them is the most obvious is to look at outbreaks, right? So if there's an outbreak and people have, gastroenteritis and they culture salmonella, and we take all the salmonella isolates and we sequence them and we show they're the same, that, I guess, corroborates your epidemiological analyses of a point source outbreak. That's the most immediate and obvious way that we would use genomics. But there are other ways that we can do it. So we can do genomic surveillance where we sequence everything and we look for changes in the pathogen. We can do source attribution work. So, if there is an outbreak, then if we can also obtain environmental isolates, food isolates, etc., then we can start to do some source attribution work as well. Similarly, with traceback investigations, we can also look at where different lineages come from. So where they evolved from. Did they come from different parts of the world? Did they, like Shigella, did they emerge in response to selection pressure? So I'll give you a couple of examples of that. So the first example is an outbreak of Yersiniosis. So some of you, I don't know if anybody here is from New Zealand, but if you may be familiar with this outbreak. So Yersinia is not nationally notifiable in Australia, but some jurisdictions Yersiniosis is notifiable, but it is nationally notifiable in New Zealand. And before I came over to Australia quite a few years ago now, I used to work at ESR, which is a national public health reference laboratory in New Zealand, and we did quite a bit of work for this there. So Yersinia pseudotuberculosis, it's a zoonotic pathogen, it's a gram-negative pathogen, and like most zoonotic pathogens, transmission is zoonotic Enterobacteriaceae anyway, transmission is fecal-oral, and infection can occur by ingesting contaminated food or contaminated water, or alternatively by direct contact with an infected human or an animal. And point source outbreaks of Yersinia are well documented. So this was an outbreak that occurred in I keep wanting to say summer, it's my northern hemisphere, I keep wanting to say summer, but I guess the spring of 2014. It never gets better. Anyway, in August, September, October of 2014, a classic, I guess, point source outbreak curve, and it was a massive outbreak, right? And these are only confirmed cases. There were 220 confirmed cases, which, as you will know, is generally only, the very tip of the iceberg, or the iceberg lettuce, I guess, in this case. Sorry. Anyway, but there were 220 confirmed cases here. The yes, unknown, and no represents patients who were hospitalized. So about a third of patients were actually hospitalized as part of this outbreak. And a number of those patients actually had surgery. Does anybody know why they might have had surgery in a Yersiniosis outbreak?
I think it was appendix.
Yeah, that's exactly right. So Yersinia actually has a tropism for the ileocecal lymph nodes. So it can proliferate there, it can cause expansion of those, and it can mimic appendicitis. So a number of patients here who had surgery actually had their appendix out, and in some of them, the specimens were sent to the lab and they grew Yersinia pseudotuberculosis. But anyway, it was a massive outbreak, and one of the features of the outbreak was that it wasn't localized to one city in New Zealand, it was actually across all major urban centres in New Zealand, across the North and the South Island as well. And so when you look at that curve and you look at where it's affected, you actually think, okay, well, that's a massive point source outbreak, and it went through some of the major food distributors in New Zealand, the contaminated produce, although I have to say nobody really knew, the source for it was actually never found. So there was some field work that was literally in the field, I think, work that was done looking at the potential source for this, but no actual source was ever named, I guess. What came out very strongly in a case control study that was never published was that it was actually bagged lettuce that came out very strongly as an association here. Anyway, usual, I guess, story with a foodborne disease outbreak hits the media, and often a source is found, but here no source was found. So at the time, what we did was pulse field gel electrophoresis, and this is a horrible laboratory method, I have to say, and it involves taking DNA from bacteria, chopping it up with not literally chopping it up, but applying some restriction enzymes to it to break the DNA up, and then running everything out on a gel and comparing it. And you literally compare it using the naked eye. So you look at it and go, okay, well, that one looks similar to that one, and that one looks similar to that one, and so they're probably the same. It's not ideal. And so what we wanted to do was to use genomics. So we sequenced as many of the outbreak isolates as we could. And again, you'll see here, and again, I'm not going to go through this in a huge amount of detail, but you can see here that this one here, remember the no real lines here, this was the outbreak clade. So very, very consistent with the point source outbreak. So these ones here, what we also did was to classify ones that were epidemiologically thought to be part of the outbreak, but actually when we used genomics, they weren't part of the outbreak, but they still had that same pulse field gel profile. So what that meant actually during the outbreak was that there were quite a few cases where the public health units had put a lot of time and effort into following up cases, but they were just the normal sporadic isolations of Yersinia that you get. So you can imagine that when you've got finite public health resource, you want to be able to divert it to cases that you know are part of the outbreak. And so I think, if we use genomics in real time, it really would have helped this outbreak. Okay, so based on the genomic data, there were a number of sporadic cases during the outbreak that were classified as being part of the outbreak but weren't. And we found no evidence of genetic variation across space and time. And so that really helped nail the fact that this was a massive point source contamination. All right, I'm just going to talk a little bit about another foodborne disease that we've been looking at quite recently, and that's Shigellosis-summary producing E. coli in Australia. And so this is, I'm not going to talk about a point source outbreak here, but I'm going to talk about how we've used genomics to understand where different, lineages might have come from. So what is STEC? Well, essentially it's E. coli that's taken up a phage, another bit of mobile DNA that encodes these genes, STX1 and or STX2. And you'll know that STEC infection can be associated with hemolytic uremic syndrome, so sometimes it can cause very, very severe disease. But in general, it usually causes mild to severe bloody diarrhea. And the main reservoirs are healthy ruminants, so particularly cattle. So what we did, again, we receive all STEC, actually we receive all STEC isolates in Australia at MDU, so we provide national services, or national characterization of STEC here. And we sequenced, over 500 STEC isolates over a decade. And the reason that we did that was because we wanted to understand whether STEC in Australia was distinct from STEC around the rest of the world, whether we had our own, populations here. And why is that important? Well, if you do this sort of stuff in real time and you've got a retrospective collection, then sometimes you can, if you identify, a link between something that's historic and something that you look at prospectively, then it means that you can go back and investigate that and see if they are linked or where it might come from, again, guiding your public health investigation. So when we compared our STEC isolates in Australia, to the UK and the USA, we found that they were pretty distinct, actually. The main lineage was STEC 0157, and another way that you can look at genomic data is that you can look at the mutation rate of the genome. So bacteria mutate depending on which bacteria it is and what selection pressure they're under. And when you do that, you can apply, again, some more mathematical models to look at when different lineages might have emerged. And so what we found here was that STEC 0157 emerged in Australia somewhere around the mid-19th century. And then if you think about historic migration patterns, then that's generally consistent with large-scale European immigration to Australia. And actually, if you look right back at historic records, you can see that, on the ships I guess, around that time and in the 30 or so years preceding that, a lot of cattle came over as well from the Northern Hemisphere. And so we think that that is where STEC 0157, the major lineage now in Australia, was imported from the Northern Hemisphere around that time. So that's, again, just another example of how you can use the genomic data. So you can use it immediately to look at outbreaks, but you can also, I guess, look back in time and work out where things have come from. All right, finally, I want to talk about an environmental point source outbreak that we looked at a couple of years ago now. And this is a global outbreak, excuse me, of a non-tuberculous Mycobacterium. So something called Mycobacterium chimaera. You might have heard about this. So this was the bug that was found in the heater-cooler units associated with cardiothoracic surgery. It caused, for the handful of infections that we saw in Australia, and not that many infections globally, it caused obviously a disproportionate amount of work for laboratories and public health units. And so a number of studies reported patients with post-cardiac surgery infections due to this rare mycobacterial species, Mycobacterium chimaera. And infections have been publicly reported now in at least 12 different countries, including, I think we're up to 7 now in Australia. And already there are epidemiological red flags that are raised here. Rare pathogen, patients that have had a common procedure, and all of the patients had undergone cardiac bypass surgery with a heater-cooler unit that was produced by the same manufacturer from the same manufacturing site. Okay, so I would just say that this is not something, I'm not going to say that genomics solved this, but it certainly helped to, I guess, corroborate what was strongly suspected epidemiologically. And so what are heater-cooler units? So this is what they used to look like. Now they've got a sort of fitting around them. But essentially the patient's blood doesn't come into contact with anything in the unit, but it's essentially I almost think of it as like a reverse air conditioner, if you like, where the patient's blood goes through a circuit there and is heated and cooled during surgery. And there are 2 water tanks down at the bottom there. And the water, or the sort of vents out. And intuitively, you'd think that that's not a good idea to have in a sterile theatre, but nevertheless, they were in sterile theatres. And so it sort of vents out across the sterile field. Yeah. Anyway, as I say, they don't look like that anymore. So we were first, I guess, alerted to this actually from another jurisdiction who had started culturing water from the heater-cooler units and had found this pathogen. And then there was a report from Switzerland around the fact that they'd also found this as well. And so through PHLN, what we did was to try and make sure every jurisdiction was culturing their heater-cooler units for this pathogen. And then we wanted to look at all the pathogens across Australia. And actually there was a site, 5 sites actually in New Zealand that were using those heater-cooler units and were culturing as well. So we wanted to get all of the units, all of the isolates from those units and sequence them to see whether they were the same. And we were asked by public health units across the country, are the isolates in WA related to the isolates in Victoria? And are they related to, the ones in Auckland, for example? And then around the same time, there was a patient who had a confirmed an infection with M. chimaera. And so we didn't know whether that was, sometimes you get M. chimaera infections from the environment, and they're not the same as this particular M. chimaera, but you can't tell that without using genomics. So there was a patient who had an M. chimaera infection, and also there were isolates from the heater-cooler unit from the surgery that the patient had had. So a lot of questions were being asked of us, I guess, as a public health reference laboratory. Are the isolates around the country the same? Are they the same as the ones that have been seen in the Northern Hemisphere? And has that patient got a heater-cooler unit associated M. chimaera infection? So we gathered up all the isolates, and this is what we found. So we got a number of M. chimaera isolates that weren't associated with heater-cooler units, But these ones here in this clade, the G1 clade, these are the ones that were associated with heterocooler units here. And yes, isolates from WA were the same as isolates from South Australia, same as isolates from Victoria. And this box here shows the patient. So this is the patient isolate here, and these two isolates are the ones from the heterocooler unit. And this was the first time that it had been demonstrated unequivocally that the heterocooler unit isolates were causing patient infections. And around this time, we got contacted by the CDC in Atlanta because they were looking at this they'd seen an outbreak in Pennsylvania in a healthcare facility there. And so they were interested to know whether the isolates in Australia and New Zealand were the same as the isolates in the Northern Hemisphere. And again, this comes back to the beauty of genomic data, is that it's universal and it's transferable. So again, we had a teleconference with them, probably about I think it was about 8:30 in the morning. And by 1 o'clock that afternoon, we had said, send us your data and we'll compare it to ours. And by 1 o'clock, we were able to tell them their data's in the stars here, that yes, they were linked to the isolates from Australia and New Zealand, confirming a global outbreak of this pathogen. One of the things that we also did with this data is that another use of genomic data is that you don't just have to look at relatedness of things, you can look at the difference between different pathogens as well. So what we did was we took our heterocooler unit associated M. chimaera and we compared it to a whole range of other mycobacteria. So Mycobacterium avium intracellulare, even Mycobacterium TB. And what we wanted to do was to find bits of DNA that were very specific to that heterocooler unit associated M. chimaera. Why would we want to do this? Because we wanted to develop a diagnostic test for this particular pathogen, and we did. And so we have been using this to look at water samples from heterocooler units to see whether or not they contain M. chimaera or not. The reason for doing this is that M. chimaera literally takes weeks and weeks and weeks to grow. But this test is very useful as a quick way of ruling in and out. We have tried it, it's not a validated test, but we have tried it on some patient samples as well with some good results actually. All right, so just to wrap up, where is this going? So if you are still on the ball at 4:45 on Saturday, what you will have noticed is that all of the stuff that I've talked about has been retrospective. So there have been things where we've gone back and we've looked at isolates when we already know or we suspect that there has been a problem. But the future for this is prospective, is rolling it into real-time surveillance, doing real-time analysis, and acting on the results. And I guess from a laboratory perspective, there's only so much that we can do in the lab. The bottleneck, I think, will be in affecting a response from the data, because there's a lot of data. So what does that mean? That means that there's workforce training and retraining issues as well about how to use and interpret the data. And that is not just in the laboratory, that's out here as well. The labs who do this need infrastructure, they need software, they need hardware to do this. You also need to make sure that the labs are all doing the same thing, right? And that we're reporting things in the same way. So, there's no point us looking at data and applying different thresholds, different tools in Victoria if New South Wales or Queensland are doing totally different things. That is absolutely not helpful for national response. And as I said, there is a lot of data being generated and we need somewhere to store that. We need to work out better ways of sharing data. I can't tell you how difficult it is to share data across state borders. Yep. And so these are all things that we're working around. And I guess one of the things that we're doing to try and overcome this is that we established something called the Communicable Diseases Genomics Network about 4 years ago now, which we coordinate at MDU, but every jurisdiction has representation on that network, and it's been very useful. We've run a number of symposia and workshops now for not just for laboratory staff, but for public health staff as well. In communicable diseases genomics, we're working through standardization of this method across different labs, and we're trying to set up a national framework for data sharing and storage as well. And we've done a number of reports to Commonwealth on this, and as I say, run a number of symposia as well. Okay, so just to summarise, genomics will absolutely transform infectious diseases epidemiology, and in fact I already think it has. There's no going back from this technology, and there's absolutely no way that you can have a career in public health without knowing about it. I think that's fair to say. It's increasingly being used to influence public health practice and policy, but I would just say that if I just put a tree up there and I had no metadata at all, I'm not saying it would be useless, but it's certainly the synergy comes from when you can link it to very good quality epidemiological data, and that requires a strong partnership between the laboratory and between public health units as well. And as I hope you can see now, there's huge potential for this data, not just for public health as well, but obviously for academia and for generating hypotheses about future studies as well. And a lot of acknowledgements, particularly the Victorian Department of Health. I think they are extremely forward-looking in their use and implementation of this technology.